discussion we talked about at the break. Tonight we have a special treat. We have two speakers, both undergraduate students at Oakland University. The first of which, Tony Coleman, is going to give our short presentation tonight. He's a biochemistry major. Okay, so get your chemistry book out and dust it off. He is currently doing research on evolutionary biology. Plans to continue his education after his bachelor's degree by pursuing a Ph.D. in astrobiology. His presentation is entitled, and I'm not even sure I know how to pronounce it. Oh, it's in a different name. Yeah. Building a large phylogeny of Archaea. Okay, so I'm looking forward to hearing this. I hope you are too.
So as you said, I'm an undergrad, Oakland University. I've been doing this research for about the past year, and we'll jump right into it. So prokaryotic life is broken up into two major groups. There's bacteria and archaea, both single-celled organisms. For my research, we chose to focus on 5 of the major phyla, or different groups of archaea. And as you can see here, 2 of the groups, the Archaea and the Archaea, they're relatively well sequenced with quite a few fully sequenced genomes available to work with these other 3 groups lacking in that department. And that is a problem in the bioinformatics field with these genomes. The research is pushing ahead of what's available to look at. So looking at that problem, we decided to look at how the evolutionary rates within these sequenced genomes affect the accuracy of building these phylogenetic trees, or you can think of it as a family tree. So here, don't get too overwhelmed. This is just a slide of some previous research we chose to compare our results to. The main takeaway here is that everything's very different. There isn't a consensus between what's been done previously with previous data and the advent of more data. Things are still fuzzy. There isn't a clear picture of how all of these 5 groups are related to each other. So moving into how I did things in the lab is we had 135 fully sequenced genomes, and this right here, this orthologous group, that is a gene that is shared between all of the 135 species. So there was 50 different genes shared between all of these 135 species. And then we built what was known as a maximum likelihood tree, just a type of tree common to the bioinformatics area. And again, you guys aren't very worried about this, but this is the LG model we ran a program to figure out the best way to analyze it as people in different areas create different programs and come up with different models to analyze this stuff. And that's another sticking point of how to actually look at these trees and build them. So this is what we used. And an interesting thing we discovered is on this graph you see what is known as the sum of branch length value, and that is a way to estimate our evolutionary rates of the genes that we were looking at. And on the x-axis is just each gene. So when looking at this, we were able to pick out the slowest evolving of our genes and the fastest evolving of our genes, which you'll see what we did with them in a minute. So jumping into the results of what I looked at, you see that 37 out of the 50 different genes that we looked kept what is known as a monophyly of the Archaea and Archaea, meaning that each of the species within these groups that we looked at stayed together, and that's known as monophyletic, whereas 13 of the ones that we looked at split up these groups, which was concerning. So when looking at these 37, we looked at to see if there was a consensus between them, and of course there wasn't, because why would science work the first time? So in order to strengthen our signal within our data, we decided to run a concatenation or built a one long giant sequence just chained together of all of our 50 genes. And then, as I said before, we built 2 separate concatenations of the slowest evolving genes that I showed you and the fastest evolving genes to see what discrepancies we might get, if any. So here's the results of our full concatenation tree. Now, don't let these triangles freak you out. The triangles are simply the number of species within each group. So this large triangle here is the Archaea. They have the most number of species. There's no correlation with how things come together. And these numbers right here are what are known as bootstrap values, or confidence level, you can think of it as. So right here, we're 100% confident that these 2 groups are sisters. And so this is the results of our full concatenation. You know, you look at it, see the pretty colors, see how things are separated. And now compare that to our slow evolving tree, the results of this. First of all, I'd like to point out our confidence levels have decreased with the less data. When you use less data, the confidence levels have a tendency to decrease, which again causes problems. There's, I swear, more problems than answers in this field. But a key takeaway is that these have been kept as monophyletic, they stick together, and so previously you see that the core Archaea are clustering here with the crown, and in the slow tree, with it's a slow confidence value, but you see that now this different group, the Talma Archaea, is seen as related to the crown. So no real consensus there. And then this brings me to our fast evolving tree. And so when I was talking about the monophyletic groups, you see that the Archaea group and the 2 previous trees was kept together, has been broken up into 3 different parts. So that was really concerning, and it showed there was an obvious effect of the evolutionary rate on the building of these trees. And it did the same thing as well, broke it up into 3 separate groups for the Archaea, which in all the literature we looked at, they're really well-established groups that should be resolved as related. So with that in mind, we decided to look at the 50 individual trees to see if there was any relation between the evolutionary rate and how confident we were in the positions of the 3 relatively unknown groups. And as you can see from these scatter plots here, there is no correlation. You know, if I were to put our values up there, it would be right around 0. So moving on from that, we then chose to this study by Phylogeny et al. Was a landmark study that had just come out in May and kind of burst our bubble with the research. But we decided to use that to compare our results, and we see that this study here, they analyzed evolutionary rates as well, but they did it slightly different than us. We removed whole genes. They had removed fast evolving sites or individual portions of the genes. And we found that we got a similar tree compared to theirs, but this full concatenation here includes both of our slowest evolving genes and fastest evolving genes. And the slow one is kind of doing its own thing. So, conclusions that we were able to reach is that the evolutionary rate has an obvious effect on the backbone of these Archaea family trees. It even affects the clustering within these two major groups that are supposed to be seen as monophyletic. And removing fast-evolving sites leads to a similar phylogeny of one that uses slow and fast-evolving genes. So why do you guys care about this? I'm doing this research to look into, I think that in order to look for life outside of Earth, which is what I want to do, it is imperative that you first learn how life came to happen here on Earth. And building these phylogenies allows us to see how the Archaea related and maybe allow us to trace back to Luca, or the last universal common ancestor, and see traits that popped up there, so we can maybe draw the line between the shirt I'm wearing, the Miller-Urey experiment, where we got this prebiotic soup to what started life, and maybe start looking for signs of that on other exoplanets, although I have no confidence we'll ever reach them outside of our solar system. A big thank you for doing this research is obviously my mentor at Oakland University, Dr. Fabio Bonastuzzi, who got her Ph.D. in astrobiology from Penn State, and Oakland University for providing the lab, and this one was for when I went to Chicago. They provided me a travel grant. So yeah, if you have any questions, I know this is very far from astronomy related, but I'd love to talk about it more.
I think we have a question down here.
We'll just take a couple of questions.
Well,
Yes, Dick?
what is the source of these various species? Are they bacteria? Are they algae?
So when you say they're single-cell organisms that have just been cultured, Archaea, it's one of the three main branches of life, where there's Archaea, then there's the bacteria, and then eukaryotes, which is all plants, animals, things like that. And they're typically extremophiles, live in like the sulfur pits, and some of these guys even feed off of rocks. They're ferrous-loving, iron-loving things. So they're the extreme ones, which we think we have a better chance of finding out in space, which is a more extreme environment.
Are they found in plants, animals?
No. Yeah, they're actually, you have a ton of them in your stomach right now. That's what produces the methanogen, which makes you fart. They feed off of what you eat. So.
Okay, if there are more questions, let's do them one-on-one during the break, and let's thank our speaker again.